In this lecture, I'm going to discuss with you an alternative method to identify cell signaling pathways from differentially expressed genes. The method is called Expression2Kinases. So first I would like to present to you a definition for a cell signaling pathway. We encounter cell signaling pathways in several of the previous lectures. For example, when we did KEGG enrichment analysis, or when we looked at the cell signaling pathway diagrams from science signaling or the KEGG signaling pathway database. However, we have to keep in mind that, as we've seen for the KEGG pathways share components, so they overlap. And also, as was mentioned by the lecture about networks, many of those pathways are connected to each other, forming a large cell signaling network. I attended a workshop at Johns Hopkins, and half of the workshop was spent on trying to define what is a pathway, and specifically what is a pathway of toxicity. And I offered during the discussions the following definition that was adapted into a review article that is going to be published soon in the journal Alttex. So what I suggested for defining a pathway is that a pathway is a warm, fuzzy cloud. So it is warm because we are close to the answer, but we don't know exactly all the members of the pathway. It is fuzzy because the memberships of components, typically proteins, within pathways is graded, meaning that some are more critical for the pathway and other components are more accessories in the pathway. And it is a cloud since the boundaries for pathways are not sharply defined, because all those pathways are connected to each other. However, the definition of a pathway is very helpful for us to organize and understand how cell signaling is initiated and executed. One of the first projects that we've done in the Mayan lab, when the lab was established, was to collect kinase substrate interactions from various available online databases to create a large-scale network of kinase substrate interactions, as well as a gene set library where the terms are the kinases and the substrates of each kinase are the sets in each row in the library. So now we can use this gene set library to perform enrichment analysis, where we give the program a set of proteins that are potentially differentially phosphorylated, and the program can prioritize the potential kinases that are responsible for those differential phosphorylations. And this typically is achieved to identify the differentially phosphorylated proteins, a method called SILAC phosphoproteomics that can be used to identify changes in phosphorylation levels on proteins. The method uses an unspecific antibody that binds to many phosphorylation sites on many proteins, and those proteins get pulled by the antibody, and then using mass spectrometry, those amino acid fragments that came off with the antibody are sequenced, and then the proteins that they belong to are identified. So we implemented a web-based tool called KIA, Kinase Enrichment Analysis, where users can cut and paste their sets of differentially phosphorylated proteins and then obtain a ranked list of kinases and also a network of kinases that potentially phosphorylate the differentially phosphorylated proteins. We also use this approach, as well as genes to networks, to analyze data collected from Robert Lefkowitz's laboratory at Duke, where Kevin Zhao, who is a postdoc in Robert Lefkowitz's lab, performed SILAC phosphoproteomics as well as beta-arrestin IPMS experiments to identify both differentially phosphorylated proteins and binders of beta-arrestin after stimulating angiotensin receptors with a biased ligand that signals through beta-arrestin. So together with Kevin, we generated this figure for a paper that was published in 2010. And when Bob received the Nobel Prize last year, he presented this figure as a part of his acceptance speech. So let's just look at the short segment of his acceptance speech that talks about how the cell signaling network downstream of this receptor is gradually becoming unraveled.
Now, I mentioned to you just a couple of minutes ago that one of the major consequences of Berestin recruitment is to lead to signaling in a fashion which is parallel to, and in many cases completely independent of G proteins. So, we were interested in obtaining and gaining a more global view of what signaling due to Berestins might be like. And so Kevin Zhao in the lab used mass spectrometry and proteomics techniques to map essentially all phosphorylation reactions that occurred in a cell when the angiotensin receptor was activated by a purely Berestin-biased ligand, such as I showed you, which can't activate G proteins. And I don't have time to go through it, but I simply want to show you that in this very diffuse map, you can see that there's a huge amount of signaling that goes on, all these phosphorylation reactions, many of which are very similar to or identical to ones which can be activated through G proteins. This tells us several things. One, Berestin-mediated signaling is very diverse and maybe every bit as diverse as G protein signaling. And two, many of the same pathways are activated. However, almost invariably, when the same pathway is activated, there are distinct physiological consequences to the different subcellular distributions of the activated enzyme.
The next project in our lab, after we developed KIA and Genes2Networks, was a similar effort, but now we collected data or generated a gene set library for transcription factors and their putative targets from studies that are called ChIP-seq. With ChIP-seq, you can identify where specific proteins bind on the DNA, and in particular we're interested in transcription factors in mammalian cells and their putative targets. So many individual studies are focused on one particular transcription factor, and then using ChIP-seq, they identify where the transcription factor binds. So we decided to collect many of those studies and generate a gene set library where the term is a transcription factor from a particular publication and a particular experiment, and then the genes in the set are the target of that transcription factor. So far we are able to identify 226 publications that contain information about the binding of 202 unique transcription factors. They are estimated to be about 1,000 transcription factors encoded in the human genome. The target genes in the CHIA database cover the entire mouse and human genome. On average, a transcription factor has over 1,000 putative targets, and the total number of interactions in the database so far is reaching 500,000. So now with this tool, you can enter a set of differentially expressed genes, and then you can potentially identify the most likely transcription factors that are regulating the differences in expression of those genes, using, for example, the Fisher test or more sophisticated tests, such as the gene set enrichment analysis test that Neil is going to describe to you in detail. Expression2Kinases mesh the three programs, CHIA, Genes2Networks, and KIA. So we take a set of differentially expressed genes. We have the up and down genes. These could be genes differentially expressed in a specific disease condition, or genes that are differentially expressed after perturbing a cell with a drug or ligand, or knocking down a gene within the cell. And then the next step would be to identify the transcription factors potentially regulating those differentially expressed genes. So we can come up with a set of, let's say, 5 or 10 of the transcription factors that are most likely to be the regulators of those differentially expressed genes. Then using Genes2Networks or using protein-protein interactions, we can find connections between those transcription factors to build a subnetwork of protein-protein interactions that is centered around those transcription factors. Then we can take that subnetwork of proteins and then convert it to a list of proteins and feed it into the KIA module, or the KIA gene set enrichment analysis process, and identify the kinases that are most likely regulating those proteins. Within this process, we recover a potential pathway from the kinases to the differentially expressed genes. So we use that process for a study that was published in 2012 in collaboration with Sijang He, who is a physician scientist at Mount Sinai, and is studying kidney disease. Sijang's area of research also involved studying HIVAN, which is HIV-associated nephropathy. Many patients with HIV develop kidney disease, and this kidney disease is caused by the introduction of HIV proteins in kidney cells. For example, when HIV proteins are expressed in podocytes, which are the cells that have food processes, and those cells, they have those food processes that set up a sieve that does not allow proteins to enter the urine. So what happens in IVAN, those cells, those podocytes, they undergo de-differentiation. So they retract their food processes and become round, and then those cells start proliferating. And this is what you see in this microscopy image on this slide. So the glomeruli collapses and no longer can filter proteins, so you get what is called proteinuria. You get proteins in your urine. So in order to study the disease, there is a transgenic mouse, which is a mouse that has in its genome HIV proteins that are expressed. You cannot infect mice with HIV, but you can program mice by inserting into their genome HIV proteins, and this is a transgenic mouse that has several HIV proteins, and those mice develop kidney disease that look very similar to the disease that is also seen in humans. So Sijang profiled kidney cells from those mice, those collapsing glomeruli kidney cells, using gene expression microarrays, and compared the expression of genes in those mice to wild-type mice with healthy kidneys. He also performed protein DNA arrays, which are a way to measure transcription factor activity. So using those two approaches, we first identify the transcription factors that are most likely regulating the differentially expressed genes seen by the microarray, as well as appearing to be differentially activated using the protein DNA arrays. We then took this list of transcription factors and connected them using known protein-protein interactions using the program Genes2Networks. And then by applying the kinase enrichment analysis on the list of all the proteins in this subnetwork, we prioritized potential kinases that could be implicated in the disease. So the first two kinases that resulted from this analysis were MAP kinase 1 and MAP kinase 3, and those are also called ERK1 and ERK2, and those kinases were already known to be upregulated in the disease state. However, the third kinase, HIPK2, has not been implicated in the disease, and it's less studied. And Sijang went on to show that HIPK2 is actually important for the disease progression. If you cross mice that have HIPK2 missing or knockout HIPK2 mice with the TG26 mice, these crossed mice do not develop very severe kidney disease as it's seen in the TG26 mice. Also in cells, so Sijang also performed several cell-based assays to show that HIPK2 contributes to the progression of the collapse of the glomeruli and the de-differentiation of podocytes. So HIPK2 was identified as a potential drug target that can help people that develop this disease. As I've shown you before in the Genes to Networks lecture, we developed a desktop application called Expression2Kinases, where you can perform this analysis yourself without too much effort. So let me demonstrate this Expression2Kinase tool again, but now we will perform this Expression2Kinases analysis. So first, let's open the desktop application. This time we're going to use the first tab called X2K analysis. It has 413 genes. I'm going to paste it in this input box, and I'm going to specify the output directory. I'm going to call it X2K demo 2, and I'm going to save. I can change the colors of the nodes. For example, I can make the transcription factor nodes magenta, the intermediate nodes I'll make those gray, and the kinases I'll keep them to be green. I can also make a network for cytoscape and Paget. I can also specify, for example, how many transcription factors I want to connect after identifying the top transcription factors. So I can change that to 8. I can use the CHIA database or some other methods to identify the transcription factors. And I can use human and mouse, or just mouse or human. And there are 3 methods for scoring. You can read about those in the Expression2Kinases paper. Then for the protein-protein interactions, we have many settings. And then for the kinases, we also have very similar settings. As I showed you with CHIA, I'm going to change this to the top 5 kinases. Now if I go back to the X2K analysis tab, I click Start Analysis. First, the program identified the transcription factors, then protein network, and then the kinases. Again, to look at the results, the best thing is to go to this directory. So this is the network. You see the YED icon. I can click on that YED network, and this opens up the network in YED. Again, I'm going to change the layout to organic, and I'm going to press OK, and it generated a network. So now the transcription factors are shown in magenta, the kinases are green, and the intermediates are gray. And this also looks like a MAP kinase cell signaling pathway, where the 2 major transcription factors downstream of those kinases are MYC and E2F1. The size of the nodes is indicated based on the number of links the node has in the network. One tip that I would like to give you about YED, you want to go to File, and then in Preferences and under Display, you want to make sure that Paint Node Over Edges is selected. So how do we know that Expression2Kinases really works? Did we just get lucky with our collaboration with CJANG? Is it going to work for all types of conditions, cell types? So this is a tough question to answer, but we were able to make headway trying to validate Expression2Kinases at a global scale. And for this, we use the connectivity map database. So the connectivity map is a grand effort that started at the Broad Institute, where they performed over 6,000 microarray experiments, and the experiments were to treat various human cancer cell lines with over 1,000 drugs. Most of those drugs were FDA-approved drugs, and then measure gene expression changes after 6 hours. And this was published in a science paper in 2006. There is going to be a new version of the connectivity map with many more perturbations, and I'm going to discuss this new version of the connectivity map later on in the course. So using the CMAP data of differentially expressed genes after various drug perturbations, we try to see if Expression2Kinases can recover the drug targets from the differentially expressed genes by reconstructing the pathways upstream of the up and down differentially expressed genes. So the first thing that we've done we identify the 500 genes that increase or decrease in expression for each of those drug treatments. In order to identify the drug targets, we needed to know for each drug which are the targets of the drugs. And this information we were able to obtain from a very useful database called Drug Bank. So this is the results from our validation effort, and I'm going to go through this slowly. So let's go from the leftmost part of the slide towards the right. So if you take 500 randomly selected genes, you will encounter a drug target for a specific drug about 2.6% of the time. If you take this same 500 randomly picked genes and select a drug randomly, and then from those 500 randomly picked genes, you build an expression-to-kinases pathway by finding the transcription factors and then connecting those transcription factors and then finding the kinases. You will identify targets within those pathways 7% of the time. And this is because drug targets are well studied, and there is a higher chance that they will appear in the protein-protein interaction networks. If you take the 500 differentially expressed genes that were differentially expressed by the drugs from CMAP, but you shuffle the actual drug, so now you're looking for the wrong targets, you still will get 8% of the time the correct target by accident. And this is because those are not randomly selected genes, those are genes that are differentially expressed due to the drug perturbation. And then you can repeat this using either the transfac prediction or the chia prediction for the transcription factors. And it looks like transfac is recovering more drug targets. One thing that Pierre Bourque already showed was that within the differentially expressed genes, you will find drug targets much more commonly than by chance. And this is because the cell tried to compensate for either the loss or gain of the target, and sometimes the cell downregulates the target, and sometimes the cell would try to compensate and upregulate the targets. So most of the time the cell will try to upregulate the target, and most drugs inhibit the activity of a downstream proteins. So 12.53% of the time, you will find the targets in the 500 up differentially expressed genes, and 10.74% of the times you will find the targets in the 500 downregulated genes. What a lot of people are doing is they take the sets of differentially expressed genes and they build a protein-protein interactions that connect the mRNAs. So this is a typical exercise trying to identify pathways by connecting the mRNAs of the differentially expressed genes. And this actually works to a certain limitation, and you can see that when we try to do that, where we take the mRNAs and feed them into genes to networks, you will find that drug targets 12.5% when you create a protein-protein interaction network directly from the upregulated genes, and 13.58% when you create a protein-protein interaction network from the downregulated genes. However, if you use the expression-to-kinases pipeline, you can go all the way up to 17.5% of the time recovering the correct drug target. And this is without playing around with the various settings. Just using the default settings in Expression2Kinases, we show that you can potentially recover the correct pathways more accurately.